Mitosis in the cell cycle. Reading comprehension activity. Have you ever looked down at your arm or leg and were surprised to see a cut that you had no idea you had? You probably noticed, or you probably placed a bandage on it, and in a few days it was healed. Did you think about how your cut healed? Have you ever wondered about why you are a lot bigger than you were in kindergarten? Have you ever looked at your fingernails and noticed they were a lot longer than last time you trimmed them? What do these instances have in common? The answer is mitosis. Mitosis is a type of cell duplication that is completed by your somatic cells to make new identical cells. Somatic cells are cells in the body that are not gametes, sperm or egg, or cells that are not used to directly form a new organism during reproduction. Non-somatic cells undergo cell division in a different process called meiosis. So somatic cells are all your body cells except sperm and egg. Mitosis takes place in a larger process called the cell cycle. The purpose of the cycle is to duplicate somatic cells. Why duplicate? The cells in our body can get worn out, damaged, die, and need replaced. Also, when you are growing from a child to adult, more cells are needed in your body in order to grow. To ensure that we have enough of each type of cell, our body cells must duplicate through the process of mitosis. After mitosis, the number of chromosomes in the same daughter cell After mitosis, the number of chromosomes is the same in the daughter cell as it is in the parent cell. 3. Cells duplicate their genetic information in the form of chromosomes during a process called interphase, and then divide into two identical cells during mitosis. These duplicate cells are called daughter cells and are identical to and perform the same functions as the original parent cell. You wouldn't want your damaged skin cells replaced by fingernail cells. During mitosis, muscle cells make identical muscle cells, skin cells duplicate into identical skin cells, heart tissue cells duplicate into identical heart tissue cells, and so on for all types of cells. It is important to remember that the cell cycle also occurs at different rates for different types of cells. 4. Chromosomes are tightly coiled pieces of DNA and protein called histones. DNA contains our genetic information and instructions for all of life's processes and is located inside the nuclei of the cells. Humans have 46 chromosomes in their cells. Potatoes have 48, and king crabs have a whopping 208 chromosomes. The first phase of the cell cycle is interphase. During interphase, chromosomes prepare to make duplicate copies of themselves. While in interphase, a cell is growing and replicating its DNA and carrying out daily cell functions. Interphase is further divided into three subsets called G1, S1, G2. We don't need to know about the subsets. Cells spend most of their time in interphase duplicating their DNA. The nucleus is still intact during interphase. Because DNA is duplicated during interphase before mitosis, the amount of DNA in the original parent cell and the daughter cell are identical. This is interphase, prophase, metaphase, anaphase, telophase, cytokinesis. 5. After interphase, mitosis officially begins, where the cell starts to divide into two identical cells. Mitosis takes place in 4 stages before it is complete: prophase, metaphase, anaphase, and telophase. To help you remember the steps in order, you can remember the acronym PMAT. P for prophase, M for metaphase, A for anaphase, and T for telophase. You could also use the mnemonic device to remember the steps. In a mnemonic, oh geez, let me have some coffee. Hold on. In a mnemonic device, the first letter of each word represents the word to help you remember it in order. For example, pretty mice are terrifying, or Peter made a taco. What mnemonic device can you come up with for the four stages of mitosis? 6. The first step in mitosis is prophase. During prophase, DNA molecules shorten and condense or thicken to form tight coils of visible strands of chromosomes. Enzymes in the cell break down the inside of the nucleus, called the nuclear envelope, and spindle fibers form to help separate the duplicate chromosomes during mitosis. Okay. The little strings beside the thing that looks like it has hearts in it, that is spindle fibers. The next stage in mitosis is metaphase. With the help of the centromeres, and the centromeres in this picture are where the X is. It looks like the center of the X. In the next stage of mitosis, is metaphase. With the help of centromeres, the spindle fibers pull the chromosomes to the middle of the equator of the cell. At this stage, the nucleus has been completely broken down and is no longer visible. Metaphase is one of the easier steps to remember if you think M for middle, because during this phase, the chromosomes are lined up in the middle of the cell. The third phase or step in mitosis is anaphase. During this phase, the centromeres are split and the sister chromatids are pulled toward opposite poles of the cell by the spindle fibers. So you can see now it looks like, I don't even know what you could say these things look like. Little clothespins, I guess, attached to the spindle fibers, and they're being pulled to opposite sides of the cell. It helps to remember what occurs in this phase if you think A is for away, because the sister chromatids are being pulled away from one another to opposite sides of the cell. Number 9. Once anaphase is complete, the cell moves into telophase, the final phase of mitosis. During telophase, the spindle fibers disintegrate and the nuclear envelope reappears. In other words, your nucleus is forming now again around your chromosomes, surrounding the duplicated chromosomes on both sides of the cell. In this step, you can observe the two sets of identical duplicated chromosomes in opposite sides of the cell. After telophase, mitosis is technically complete. However, the two cells are still fused together by the cytoplasm. And you remember that cytoplasm is the liquid inside the cell. To completely separate the two fused cells, a process called cytokinesis splits the cytoplasm of the two cells apart and allows the cell membrane to reform inside two new identical cells. After cytokinesis is complete, the cell cycle is complete. It is important to note that mutations or mistakes during duplication can happen during mitosis. Mutations can damage DNA, cause cells to die, or even cause them to over multiply out of control. Cancer may occur when damaged cells multiply too quickly, causing serious harm to the organism. Cancer is uncontrolled cell growth caused by uncontrolled mitosis. Okay. Thank you very much.